All right. Well, thank you, Tony. It's my absolute pleasure to introduce Dr. Simon today, who received his Bachelor in Neuroscience from Princeton and then his Master's and PhD in Physiology and Biophysics from NYU. He then moved to Rockefeller for his postdoc and is never left. He is now the Gunther Blobel Professor there and leads the Laboratory of Cellular Biophysics. I think what I find really inspiring about him, his lab and his career has been his ability to focus on very basic processes within the cell. He has been able to look at single viral particle shuffling within cells, at single molecules, at single channel proteins, but also he has then moved to very translational work within the context of cancer. He works particularly on something that's near and dear to my heart as well, which is rare childhood and young adult cancers, particularly liver cancers, which we're going to hear from him today. And given, in the interest of time, I'm just going to give him the go ahead. And thank you so much for joining us, Sandy.
Alice, thank you very much for the invitation, and Tony as well. So what I'm going to tell you about today is a very specific liver cancer called fibrolamellar hepatocellular carcinoma. It was first identified in 1956. Basically, of these very large liver cells. Can people hear me? Tony, just give like a thumbs up or something. Wonderful.
We can hear you. Perfect.
Great. Mainly identified by the histopathology, but the problem is there's no standard of diagnosis. As a result, it's believed there are many, many examples of misdiagnosis, which I'm happy to talk about afterwards if people are interested. But its main characteristics are these sort of fibrous bands running through made of collagen. It mainly hits young kids, adolescents, young adults. Its peak detections at 21 years. It's officially estimated at 1 in 5 million, but as I'll show you soon, now that we have a clear molecular diagnosis, there's evidence that actually it's significantly underdiagnosed, at least in the US, 10 to 50-fold underdiagnosed. It's presented initially as a primary liver cancer, a very large mass. There's no other underlying liver disease. You don't see viral infection. You don't see cirrhosis. So in general, there's a paucity of symptoms or serum markers. It's still officially codified as a subvariant of hepatocellular carcinoma. So as a result, most patients are just treated as if they have hepatocellular carcinoma. And the problem is none of the systemic chemoimmunotherapies have been found effective, including none of them for HCC work. So essentially, the only current management is surgical resection. When we started working on this about a decade ago, what was not known is it one disease or many diseases. In other words, this is just a catch-all term for any kind of liver tumor that has fibrous bands. Is it something that's genetically inherited or somatic mutation? And the problem with working with such a rare cancer is that there's just a lack of information. There's lack of diagnostic tools. There are too few samples. Let's see if we can hide this. So the few samples is little funding, little information. But still, there are advantages to studying a rare cancer. So they're often very precisely defined, as you'll see, often quite homogeneous. And as a result, it's possible to make a lot of progress pretty quickly. So I'll just give one example. From looking at retinoblastoma, which is actually even more rare than fibrolamellar, they found the only thing that was altered was a deletion in one gene. And with a deletion in one gene, the only way to finally explain it was positing tumor suppressors. And from that, we now realize that tumor suppressors are actually much more common in cancer. So our approach was to partner with patients through social media. We established a medical registry that I'll show you about. We now have over 250 patients from over 20 countries. We have a tissue repository with tissues from 150 patients. And we did sort of a classic analysis of whole genome, transcriptome, proteome, and phosphoproteomics. So the advantage of setting up this registry with the patients is that it meant that since we have it run by the patients and the family members, that we can really follow patients over time. There are very few patients at any one institution. If, let's say, Dana-Farber gets 3 a year and Sloan Kettering's 3 a year, it's very hard to get sufficient numbers to analyze things. There are even limited numbers in any one country. And by having this run by the patients and family members, what we do is we now follow the patients as they move from institution to institution, even country to country. Equally important, we follow the patients even after their therapy is done. So we're in constant contact with them. And this allows, very long follow-up. It also means that if there's a molecular characterization down the line, we can now edit the data. In fact, we found 15% of the patients who officially diagnosed as fibrolamellar did not have it. Since it's run by the patient and family community, we guarantee the patient data won't be sold. So we get a tremendous buy-in on the parts of the patients. And we have not only all the patients' electronic health records, we also have a whole series of other questions we've come up with that we actually considered relevant to the disease. We came up with them in meetings with patients, family members, oncologists, hepatologists that are not part of the standard health record. What we do is, as encouraging patients to participate, we provide information for them about the science. So it's not just a question of getting the patients to come to the scientists, but the science to the patients. We have, for example, frequently asked questions about the disease. We have definitions of a lot of key terms. We have a little video where you can learn how to go into PubMed, how to search PubMed, how to get a PDF file. We have plain language summaries for a lot of the articles. And in some of the cases, in addition to plain language summaries, we actually have little video summaries of some of the work.
In order to confirm that the chimera actually causes fibrolamellar, the researchers edited the genes of adult mice using the new CRISPR technology.
So this is part of like a longer video explaining to the patients, the science behind some of the work. We have an IRB that oversees this. At the request of a variety of groups, we've migrated the entire database over to REDCap so people can do research. So for example, Mark Yarshin was interested in immune checkpoint inhibitors. Through the registry, we got a couple of dozen patients. We got their complete medical records scans. And what Mark did is he had one group of radiologists and oncologists survey all of the data. We actually discovered immune checkpoint inhibitors do not work very well for these patients. We found, for example, there are clusters of patients in different areas. So there's a big cluster in Minnesota, right in an area where a lot of these forever chemicals being dropped. There are clusters elsewhere. And from examining these clusters, we've now identified a particular mutational signature common to all the patients, which we think is due to environmental toxin. From being able to get a lot of data, we got information on the survival. For example, survival did not correlate with age of diagnosis, which I found surprising. There was a big difference in gender. Women did much better than men. Initially, we thought that maybe they were more aware of their bodies, diagnosed earlier, but they're all diagnosed at the same age. This led to a very interesting study now on the role of exon activation in response to the tumor. A surprising observation is that survival did not correlate with tumor size at all, nor metastases. It did correlate with a number of other tumors, either in the liver or that have metastasized outside the liver. And of particular interest to our patients, we took a large group of the patients who were told their tumors were inoperable, but then we encouraged them to go on and get a second opinion. And all those who went on to get a second opinion had surgery actually survived much better than those who did not. So in all cases, all these patients were told that the tumors were inoperable. So now let's get into the biology of it. The next problem is, how do you get samples if there's only a few in each institution? So one of the patients early on went on to YouTube, this like now a decade ago, and posted a request asking for tissues.
If I could say anything to people out there with fibrolamellar, I'd just say people are working on it, and there are people who are trying to find a cure and trying to help understand what this cancer is. And we just really need tissue samples. If I could say anything.
So this has led now to the 150 samples we have. So we did whole genome sequencing. We found basically there were very low level of single nucleotide variants. There are no obvious mutations in any of the known oncogenes. Very low level of structural variants. Very few amplification, indels, inversions. No deletions that we found that were recurrent. So then we started looking at the transcriptome, and we found over 1,000 transcripts that changed significantly here. But the changes in the transcriptome were consistent from patient to patient. So for example, collagen was, certain collagens were increased in all the patients compared to the adjacent normal. So you can look here to the 3.3 to the 6.95, but that we expected. Collagen is what's involved in driving this cancer. I'm sorry, collagen is what forms the fibrous bands. It was a mistake. We found the whole EGF receptor pathway was up. So here are some ligands to the EGF receptor pathway. So if you look, EREG is up 2 to the 6, neuregulin 2 to the 4.9. If you look at individual patients, you can see the increases are almost identical patient to patient. So now if you look at the transcriptome, you see that all of the tumors clearly segregate away from the adjacent normal tissue. And for that matter, they segregate away from all other liver cancers, away from cholangiocarcinoma, hepatocellular carcinoma, hepatoblastoma. We also found changes in the proteome were consistent patient to patient. So for example, if we do UMAP plot, you see the tumor always segregates away from the normal. Similarly, the phosphoproteome, the tumor segregates away from the normal. So this is a fairly homogeneous tumor. You can look at the proteome also by just doing Western blots. So for example, ACAPs are increased tumor compared to normal. PRKCA, which is a particular kinase, catalytic subunit protein kinase A, is increased. ERBB2 is increased in tumor compared to the normal. RORA is increased. We confirmed this at the mass spec level. We also looked at the phosphoproteome, but first, in terms of the mass spec, some proteins that increased much more than at the transcriptome, and some actually increased less than the transcriptome. So actually, the proteome was giving us very different information from the transcriptome, but the critical thing is that it was consistent from patient to patient. Likewise, the metabolome was highly consistent patient to patient. So changes that we found in gluconeogenesis and glycolysis, particularly in ammonia consumption, were highly conserved. A lot of these changes in particular were involved in ammonia metabolism, which we were particularly concerned about because a lot of our patients end up getting high levels of ammonia, confusion, which leads to coma and often death. These changes, when we ask, okay, where are they actually occurring? They were in the tumor cells, not in the adjacent stromal cells, but the changes were fairly consistent from cell to cell. So it wasn't as if just a few tumor cells were increasing. It was increasing in all of the tumor cells. And if we look specifically at the genes that are involved in regulating ammonia consumption, you have roughly 70% of your ammonia being consumed in the liver. What we found is that about 35% goes through this pathway. 35%, this is carbonyl phosphate, 35% is consumed from glutamate to glutamine. This is down quite substantially. And a prediction would be that if this is down, what you should be doing is getting a buildup of these metabolites. Likewise, this pathway is down to the fifth, but the reverse pathway is up. And in fact, if we look at the metabolome, all these metabolites are increased enormously, suggesting a block of this pathway. Likewise, the glutamine is actually down quite considerably. In fact, and this is suggesting what these cells are doing, actually running in reverse. Instead of consuming ammonia, they're actually generating ammonia. In fact, if you look at the uric acid in the urine from going into the patient medical registry, we found that actually this is increased somewhere in the order of 1,000-fold in these patients. So clearly we're seeing something very consistent. It's very homogeneous, but really it's quite different from patient to patient. And actually, can you still hear me?
Yes.
Okay, good. I lost the window there. So the question is, what is going on? So our changes in the transcriptome are quite different from that of the adjacent normal. This was one exception. This was a patient that was presumably hepatocellular carcinoma segregated together with the fibrolamellar patients. We'll return to that patient. So what's going on? There were no obvious mutations that were consistent. So we did see there were a dozen kinases that were upregulated. We know of some tumors where, in fact, what you get is a change in splicing. We weren't maybe in these kinases were upregulated. Maybe there's no mutation in it, but maybe there's a change of the splicing involved. But they're using different exons. So the way you do this is you take your mRNA reads, you map them onto the DNA. So in this case, this is the the catalytic subunit of protein kinase A. It's on the negative strand, so we're reading from right to left. So here's your first exon, second exon, third exon. When we mapped the reads in the tumor for all the other kinases, they were completely normal. The exception was this catalytic subunit of protein kinase A, where exons 2, 3, 4, et cetera, were all increased. The reads between the exons were increased, but the reads between the first and second exon were not increased, and the reads of the first exon were not increased. Instead, we had reads that went upstream to a heat shock protein cofactor. And what we were seeing now is that there were still the normal reads for the heat shock protein cofactor, DNA JB1, There were still some normal reads from the catalytic subunit of protein kinase A, but now there were reads for what appeared to be a fusion transcript of the first exon of DNA JB1, followed by the rest of protein kinase A. And when we went into the patient tissue and we actually purified RNA, every single patient had this fusion transcript between DNA JB1 and PRCACA. So what was going on? Early on, we didn't see a deletion being reported. We found that every patient had a normal copy of that chromosome, but on their second chromosome, there was a deletion somewhere from the middle of the first intron of PRCACA to usually the first intron of DNA JB1, maybe a little bit into the second exon. And because of that, what you were getting a fusion in one chromosome between these two, but not in the other. We wonder, were you making a fusion protein? So if you looked in the adjacent normal tissue from each patient, you saw the normal protein kinase A, but in the tumor tissue, there was a second slower moving band, which was consistent with having this fusion to the heat shock protein, which is a little bit larger. To test if this really was a fusion protein, we used an antibody against DNA JB1 to immunoprecipitate this, then run an Argellum probe with an antibody to PRKCA, and you see this slower moving band is a true fusion protein. So the question is, there's this fusion gene and a fusion protein in every one of these patients. Is it actually sufficient to produce the tumor? So we did is we went into mice, we recreated the same deletion using CRISPR-Cas9. And when you do this, you got what appeared to the pathologist exactly like fibrolamellar. You looked at the transcriptome, it looked like fibrolamellar, and the mice died. It didn't really matter which kind of CRISPR guide you used. You'd get the same result. This doesn't distinguish whether it's due to the loss of genes in between or creation of this new fusion gene. So we just put in the fusion gene by itself with a transposon. And once again, this was all you needed to get fibrolamellar and to have the mice start dying. We've seen a similar thing since then. You can put this into primary human hepatocytes and see the same thing. So how do you use this information now to actually fight fibrolamellar? There's a few different strategies you could use. You could hit the DNA encoding it. You could hit the RNA encoding it. You could hit the fusion protein, or you can downstream elements of it. Now, to do this, you need validated model systems. So there's a few systems that we've used. You have the purified protein. We have human liver organoids made directly from patients. We have cells directly from patients, which will be a big emphasis of this work. We've implanted tumors in mice to get patient-derived xenografts. We have genetically engineered mice, and then primary human hepatocytes, into which we've now put in this altered fusion gene. Now, for evaluating these models, particularly the cellular and tissue models, you want to know, do they recapitulate the histopathology? Do they express the fusion oncotranscript? Do they express the fusion oncoprotein? Do they recapitulate the transcriptome of the patient tumors? Do they recapitulate the proteome and the phosphoproteome? And what I'll go into soon, do they actually demonstrate oncogenic addiction to this fusion protein? And do they recapitulate the drug response of patient tumors? This is very important because, as we found, is a lot of these model systems do not recapitulate these. So we have organoids. For the sake of time, I'm going to skip the data on that. What I'm going to show you is tests that we did directly on cells from patients and on our patient-derived xenografts. So I'm going to show you how we evaluated them. If you look at the histopathology of the patient-derived xenografts, they hew very closely to that of the original tumors of the patients. If we look for expression of the fusion transcript, it's maintained through all of the passages. If we look for expression of the fusion protein, so the lower band here is the native protein, the top one is the fusion protein that's maintained in all the passages. If you look now at the extent to which the transcriptome of the patient-derived xenografts, things go up or down, recapitulates that of the original patient tumors, where we're comparing the tumor to the adjacent non-transformed, you see that this really hews very closely to what we find in the patient tumors. So we have a series of these model systems that we've recapitulated. Sort of the best ones for the histopathology was the direct from patient, but the PDXs, the genetically engineered mice human hepatocytes were pretty good. They all expressed the fusion transcript. Where things deviated was in terms of recapitulating the transcriptome of the tumors, the genetically engineered mice, which now you're expressing in mice, which is a whole different set of proteins, were not quite as good as either the patient-derived xenografts, which now are really human cells, or the direct from patient screens. If you transduce non-human, non-liver cells, other immortalized human cells with a transcript, you actually have problems. The transcriptome ends up being very different. If you transduce mouse cells, it's okay, but not great. if you ask about the phosphoproteome, you start getting deviations if you do it in mouse cells, where you transduce human cells that are not liver cells. In terms of demonstrating oncogenic addiction, it completely failed if we went into mouse cells or non-human tumor cells. And there are also problems from the drug response profile. So in general, the problematic systems are the ones selected in plastic, the organoids which drift, the non-liver cells, or mouse cells. The best ones were the patient-derived xenografts, cells directly from patients, or primary human hepatocytes. So I'm going to show you how we're using these systems to actually test the efficacy and predict the efficacy of therapeutics. So for the patient-derived xenografts, or the cells directly from human tumors, we're going to dissociate the cells, distribute them into many wells of a dish, and test their efficacy against various drugs. So first, I'm going to take you through hitting the actionable targets. So the first thing we tried to hit was drugs that are in the clinic. And one of those common drugs used in this clinic is sorafenib. We've never seen any effect of sorafenib either on cells dissociated from PDXs. We've also tried PDXs growing in mice who were treated with sorafenib. Never saw any effect. For that matter, when you go back into the patient medical records, there's no sign of sorafenib working. Almost every drug every drug that's used in the clinic we saw no effect on at all. So here's a series of drugs in the clinic we've tested, romatinib on through. We don't see any effect of them either in cells taken directly from patients where we screen in the first 72 hours after resection, and no effect on cells from patient-derived xenografts. So this is just taking drugs in the clinic. So let's take a precision medicine approach. Let's now take drugs that are targeted to the specifics of fibrolamellar. So from looking at the transcriptome, we found a number of oncogenic pathways that are regulated, the EGF receptor pathway, the Wnt pathway, Aurora A. Let's try drugs against those. So if we try drugs against Aurora A, they had no effect at all. We tried a number of different Aurora inhibitors, no effect. This is on different PDXs. We've also done it in cells directly from patients, no effect. Inhibitors of the EGF receptor pathway, they had no effect at all. Actually, in a few cases, people who were concerned the PDXs may not recapitulate what happens in humans. They actually tried various clinical trials on these, and they failed in humans as well. We've done these both on cells dissociated from PDXs. We've also done these on PDXs themselves. So, for example, the Wnt pathway is upregulated. If you block the Wnt pathway, we've done this now with 12 drugs. There's no effect at all. So if you go in terms of actionable targets, in terms of the transcripts that are upregulated, none of them had any effect. So what about hitting the DNA encoding the fusion gene? One thing we considered was using CRISPR-Cas9 to just cut the DNA where this fusion is. The problem is the fusion junction is at a different location in every single patient, and it's in a region of the DNA which is quite redundant and is not very good for targeting small guide RNAs. So that actually has not been very promising. What about hitting the RNA encoding the fusion gene? So this was very important because we really want to know is this DNA JB1 Prokaka a valid therapeutic target? Is it only triggering fibrolamellar or continuing to drive it? So for example, RAS is involved in triggering certain pancreatic cancers, but you pull away the RAS and the tumors keep growing. So approach was to tile completely across the junction of the DNA JB1 Prokaka and make small hairpin RNA against them that would hit just the fusion with minimal effect either on PRKCA or the heat shock protein DNA JB1. We then made these into viruses in an inducible form. We showed that you can knock down the DNA JB1 Prokaka with very minimal effect on the native protein. We then put these inside of tumors, had them start growing in mice, and then when we induced the shRNA, the tumors not only stopped growing, but they shrunk away. And at the very end, the very few cells that left here were cells that were not successfully transduced to the shRNA. So what this says is that this DNA JB1 Prokaka is needed for continued growth of the fibrolamellar. On elimination, the cells die. The cells are oncogenically addicted to it, and it really validates this as a therapeutic target. And it says that our patient-derived xenografts are dependent upon it. If we take this same oncogene and we just put it into hepatocellular carcinoma, hepatoblastoma, so these are cells that have already transformed separately, we can induce the same shRNA, we can eliminate this oncogene, but it has no effect on growth. So if you take an already existing tumor line and just pop this in, the cells don't become oncogenically addicted. So what you really need to do this actually in the native tumor or in primary human hepatocytes that have been specifically selected to grow and depend on this. So again, the criteria that I'm going to use for all the following data is that everything we're doing recapitulates histopathology. They express the fusion transcript, the fusion oncoprotein. The transcriptome is the same as that of the patient's, the proteome, phosphoproteome. They're oncogenically addicted to this. And they show the same drug response profile, which I'm going to take you through now, also the same metabolome. So I showed you that hitting the RNA is an effective approach. We now know that hitting the fusion protein is an effective approach, but for the sake of time, I'm actually going to skip that now. Instead, one would go into what happens if you try to hit the downstream elements. And for that, what we did was a drug repurposing screen for functional precision medicine. So all these other approaches here of hitting the RNA, of hitting the protein, these are effective, but these new chemical entities, these are going to take 3 to 5 years minimum before they can go into the clinic. And the problem is there are patients who are sick now, and there are patients who are dying now. The advantage of doing this is that by repurposing drugs and finding out what actually works, one, you can get things that can go into the clinic right away, but two, what I'll show you in a moment, you're going to learn a lot of interesting things about the disease that you don't get from looking at the transcriptome. So our initial approach was to take 5,000 known drugs. Actually, we've since supplemented this with another 3,000 drugs, all of whom have passed phase 1 clinical trials. And initially, we just screened on 2 of the patient-derived xenografts, looking for any drug that had any effect at all. We then took our top hits. We added to it anything that targeted protein kinase A, anything that targeted transcripts that are increased in fibrolamellar, things in the clinic. And we tried those on 6 different patient-derived xenografts. And as a counter screen, we used primary human hepatocytes. And the hits actually were quite surprising. We found basically anything that hit the histone deacetylases was incredibly effective. I'll show you some examples of the data in a moment. That was not predicted. We found inhibitors of the anti-apoptotic pathway, BCL-XL, were extremely effective. Inhibitors of BCL2 had no effect. We found inhibitors of the topoisomerase I, irinotecan, were incredibly effective. This was very surprising. Topoisomerase I is needed when cells are rapidly dividing, and this is a very slow-growing tumor, so why would that work? We also found that napabucasin, which is inhibited both as inhibitor of STAT3 and reactive oxygen species, in EIF4A worked. So this shows you, what the screens were like. Here's napabucasin. This is now on a series of samples, in this case taken directly from patient tumors, and are showing you the counter screen here, primary human hepatocytes. We tried other agents that block STAT3 that had no effect at all, so it means it's unlikely to be through STAT3. Agents that affect reactive oxygen species had moderate effects. It turns out the company that made napabucasin was sold off. The new owners were discouraged. We've not been able to get used to the drug to push it on further. In fact, as we started testing more and more patients, we actually found there's a great deal of variability in the responses. So we're not going to pursue napabucasin any further, but it's interesting to note that other blockers of this EIF4A work extremely well. We've now done this in many more patients. This is unfortunately off patent, and it's been very hard to get a company to want to go further. So what we're going to pursue now is the irinotecan, the HDAC inhibitors, and this anti-apoptotic pathway. And one of the things we're learning is blocking that anti-apoptotic pathway actually synergizing with the other treatments. So for example, if we take panobinostat, selective inhibitors of BCL-XL synergize very nicely. So this is a plot where going upwards of things that act synergistically. Right along here are things that are additive, and things that go down here are antagonistic. Inhibitors of MCL1 synergized a little bit, and inhibitors of BCL2 do not synergize at all. Another way of looking at this is here's a dose response curve of 3 different lines to panobinostat. If we first block BCL-XL, you can see now they're now more sensitive. But if we do the same thing with primary human hepatocytes, blocking BCL-XL does not increase their sensitivity to the panobinostat. So one of our concerns in doing all this work in these patient-derived xenografts is that how do we know that these tumors that have been grown for a long time really represent the bulk of the behavior of tumors in humans? Maybe we've just subcloned out, the one rare cell when they've actually grown up in mice, where they take 9 months, a year, 2 years to grow. Maybe the tumors in patients behave differently. So all the following tests I'm going to show you are done on samples directly from patients. So here you can see the response of irinotecan. This is now in a collection of different patients. You can see they all respond very similar, but not the same. Here's the primary human hepatocytes as our counter screen. Here's panobinostat. Some patients are sensitive, and some are hypersensitive, but all are more sensitive than panobinostat. To give you a sense of the robustness of these, what I want to show is an example from a patient where we had a series of surgeries over time. So 2 surgeries that were 3 years apart. In each case, we got a number of different samples from the patient. So in this particular case, what they sent to us with 3 metastases, as well as a primary and the normal liver. And they said one of the metastases was hepatocellular carcinoma mixed with fibrolamellar. Another one they said was pure hepatocellular carcinoma. And another one they said was undetermined, was probably some focal nodular hyperplasia. They looked at, one was green, one was white, and one was red. And the oncologist, the pathologist, and surgeon all said they were different tumors. But when we went ahead and did the PCR to probe for the presence of this fusion gene in all three of them had the fusion gene in them. All 3 of them, when we then did a drug response profile, the drug response profile, in this case to the SN38, which is the active metabolite of irinotecan, was the same. Not only were they the same, if we go back to the surgery from 3 years earlier from this very same patient, they had exactly the same response to irinotecan. In fact, if you look at their response to navitoclax, the response of all 3 of these tumors were the same, and the same as the response from 2 years earlier. This is the blocker of the EIF4A. All 3 were the same. And in fact, if you go ahead and you now do spatial transcriptomics on these tumors, you see that all of these tumors in the tumor cells were indistinguishable. The difference was the stromal cells around them, and the stromal cells all had very different patterns of expression. And that's what was leading to the tumors to look different to the pathologist. Grossly, one was green, one was white, one was HCC, one was FNH. In fact, they were all fibrolamellar. They all had the same, or at least from my point of view as a non-pathologist and a non-oncologist, so you can disagree with me, they all had the same oncogenic driver, and they all had the same drug response profile. So from my point of view, I will consider them the same cancer. So now, one of the things that bothered us is that why was there a difference in a response to these patients to irinotecan? When we looked at the transcriptomes, they were almost indistinguishable between these patients. The proteome, the phosphoproteome. What was responsible for the difference? And we were wondering maybe the clue was in the levels of expression of this anti-apoptotic protein BCL-XL. So whenever we do screens directly on a patient, in addition to screening against single drugs, we screen against combinations of drugs. So in this case, for all these patients, we screen not only against irinotecan, but irinotecan combined with other agents. So what I'm going to show you now is the results of this one example here in dark blue, which is the patient who was least sensitive to irinotecan over here. When the same patient cells were incubated with increasing concentrations of navitoclax to block BCL-XL, you see you can drive the sensitivity down to single-digit nanomolar. In fact, all of these patients, when you block BCL-XL, they now show the same drug response profile to irinotecan, which means we now have a good explanation for why this variability in response. And now they're much more sensitive than primary human hepatocytes. We wanted to go ahead to use irinotecan together with navitoclax, but the problem is navitoclax has a very, very adverse effects on platelets, leading to thrombocytopenia. So the problem is, how do you get around it? So we worked with a group in developing a PROTAC based on navitoclax. So this now binds into BCL-XL, but because of the way the extension was put on, it no longer binds to BCL2. And this particular PROTAC has an E3 ligase on it, which is now used to ubiquinate the BCL-XL that is not found in platelets. So now if we treat tumors growing in mice and we look 24 hours later, we can now completely eliminate the expression of BCL-XL within the tumors. But if we look at the expression of platelets over time, there's no adverse effect on the platelets in animals that are treated jointly with our PROTAC and the irinotecan. So now, how well does this actually work on tumors? So these are now different tumors growing in mice. What you can see in the upper panel is the average. Down below are the results from a number of different PDXs. So this is just the vehicle. This is now just giving irinotecan. What you can see is you get essentially the stable disease. The tumors stop growing. Now you combine the irinotecan together with this inhibitor of BCL-XL, and not only do tumors go away, if you stop treatment here and you wait for months, it does not come back. The current status is, this is now in a phase 2 testing as a monotherapy. From going through the patient medical registry, we found 3 patients who'd received irinotecan, And in fact, for all these patients, either the tumor stopped growing or went back a little bit. So the patient had exactly the same response as we saw in these patient-derived xenografts. So we're now developing this as an investigational drug for fibrolamellar together with the Children's Oncology Group. What was sort of perplexing now is why is irinotecan working? It should only work on fast-growing tumors. So irinotecan is made in the liver to SN38, that's its active metabolite. That's what enters cells. Normally in the liver, there's a family of enzymes that puts a sugar group on it, which now exports this into the bile. It's now excreted from the body. It turns out that the kinase that's overexpressed in fibrolamellar phosphorylates the promoter for this family of enzymes. So they're actually downregulated at the protein level substantially. And because they're downregulated so far, this no longer gets the sugar group added on, so it builds up inside of the cells. So in fact, from doing this high throughput screen, we discovered this is working not because the cells are necessarily specifically sensitive to topoisomerase inhibitor. What they're sensitive to is a particular metabolic pathway for getting rid of them, which is helping us now to identify what other drugs may work. In fact, we can predict the drug sensitivity to irinotecan based on the expression level of this protein and the synergism based on the levels of expression of BCL-XL for most patients, but for a few patients also MCL1. So in doing this work, I've had a lot of people who've helped me out. When I started doing this project, I was not a cancer biologist. A lot of the liver work, primary human hepatocytes, is done by Charlie Rice and Young and members of their lab. A lot of the mitochondrial work getting going to Shai Shaham. People in the mass spec facility have been invaluable in the proteomics and for the metabolomics. With Fraser and Lavo, we did all the high throughput drug screening. A lot of metabolomics and actually the CRISPR screens I didn't go into. These are bioinformaticians who are working with us. A lot of the PDXs were developed with the help of others. Actually didn't go to the immunology work. Michelle Nussensweig, Jeff Ravitch have been incredibly helpful. I didn't go to a lot of the chemistry we're doing with Nat Gray. We've had a lot of surgeons who've been incredibly helpful throughout all of this. Mouse models were developed with Scott Lowe and Ted. A lot of the high throughput drug screening we've done together with Patrick and Tony. The work preparing for the clinical trial we're doing together with Ali O'Neill. The fibrolamellar registry, it's run by the patients, have been great. I'm sure there are other people I skipped. What I'm going to leave you with is that fibrolamellar are a result of somatic mutations, not genetically inherited. All cases are the result of a dysregulation in protein kinase A, with most of the patients having this deletion of 400 kilobases and one copy of chromosome 19. This fuses DNA JV1 to the catalytic subunit of protein kinase A, producing fusion gene, fusion protein. This is all you need to get the tumor in mice. Drugs currently in use in the clinic have no effect. Drugs that antagonize the oncogenes that are upregulated are without efficacy, but a functional precision medicine screen, Tony, this is a good lesson for you. A functional precision medicine screen identifies a number of agents that show efficacy on cells dissociated from tumors in mice, cells directly from patients, cells growing in tumors, and tumors in patients. And we can understand the efficacy of these drugs from analysis of the biology of the tumor. So why study rare cancer again? They're often precisely defined, they're homogeneous, and I think most cancers have been more precisely defined than rare cancers. Patients are eager to work with us. There's power in partnering with them. You know, the fibrolamellar patients, they're the vanguard of the battle. They've been providing for us tissue samples, blood samples. They've recruited patients, funding. They've worked in the lab. You know, so just one example of Jackson, he helped purify a lot of the protein, helped design a lot of our techniques for targeting it. His first paper just came out a few months ago, but unfortunately he died last year from fibrolamellar, as have all these other patients who are working with us. But since the patients themselves have really been taking the lead in this, a lot of the work, I'll leave to one of the patients to sort of, you know, summarize the conclusion of the work.
Rather than trying to broadly learn about all liver cancers, I just examined a small, well-defined patient group, which is what allowed for such a precise discovery. With this knowledge, we're now working on developing the first diagnostic tests and new treatments for fibrolamellar.
So the members of my lab asked me to put up a picture showing them going out and having fun. They say it's a very good way to recruit graduate students and postdocs. This isn't true. This is all done with Photoshop. They never leave the lab, but if it helps recruit students, you know, it'll work. Here are the actual people who did all the work. These are a few generations of the lab. I want to thank the private foundations invested early on, currently the NIH support. And I also want to thank all of you for your attention. And I will take any questions. What I need to do is find out how to get back into seeing the Zoom box here.
Thank you so much for the excellent presentation. So if you guys have any questions, feel free to jump in or put it in the chat if you can talk, and I would be happy to read it out. Maybe I'll just start with oh, sorry, go on, Tony.
Okay, there we go.
Great. Yeah, I have a quick question. That was just brilliant, Sandy. I love so much is great there. I mean, identifying potentially, let's hope, active therapeutics that you absolutely could not have done any other way other than just functionally treat them. And again, my mind boggles why people are resistant to this incredibly powerful empiricism. But I have a question for you, which is like, it's a fascinating observation that the reason why irinotecan is working, or SM38, it's metabolized, is working because of a downstream metabolic defect, okay? Now, we're always told that in the absence of much evidence that conventional chemotherapy works here, there, and the other places because the cells are rapidly dividing, even in the face of very indolent tumors. So I'm wondering, what do you think about this as a more, and it's also odd the way like irinotecan is used in this tumor, that tumor, the other tumor, but a whole lot not in between. And there's all sorts of examples of drugs that are used in one cancer, but not another for no good biological reason, but just because history has shown us they work. Have you thought about to what extent all of that heterogeneity has to do with metabolism?
Oh, so for example, panobinostat works incredibly well. Why should panobinostat work? Panobinostat is metabolized by CYP3A4. CYP3A4 is down enormously in fibrolamellar. And in fact, everything metabolized by CYP3A4, these cells are sensitive to. So I agree with you. I think this could be a general principle for helping at least in part describe, the sensitivity to a number of agents.
Yeah, that's great, because there's so often it's a mystery why a drug works in one tumor but not another, number one, and why isn't it killing normal cells? It's often this is just a complete mystery that even after using a drug for 50 years, we often don't know the answer.
Oh, so if we take primary human hepatocytes and we knock down UGT1A pathway, they become sensitized to irinotecan.
Yeah, perfect.
So thank you. We have a question from Patrick in the chat. I don't know, Patrick, if you wanna talk, but if not, I'm just gonna I'm gonna read it. There he is.
Sure. Yeah, I can ask a question. Great talk, and again, I'm always impressed by the level of patient engagement that you guys have done in sample acquisition. I think this is one of like many key barriers for functional precision medicine. Can you discuss some of the major challenges that you and the registry have faced in getting patient samples and how you've solved them? Or put another way, if you could do this again, how would you do it? Or like, what are the key levels?
So one of the big problems is just getting various hospitals to agree. I actually find in most cases, the surgeons are great. The surgeons want to agree. It's not clear to me what the barrier is. So for example, there was one patient who really wanted to give us a sample. We signed, we had them fill out the whole IRB, we got the consent, and the hospital involved said, oh, for us to give you this sample would involve setting up an MTA that will take roughly 4 or 5 weeks, that will cost our lawyers hundreds of thousands of dollars, and it's just not feasible. And so the general counsel's office at Rockefeller said to us, here's the MTA we've used with every other university, and it just didn't work. They just wouldn't respond. It happens that on the day of the surgery, so this is back when Obama we happened to be down in the White House again, and Obama asked my daughter, well, how's the collection going? Because we discussed from this issue of patients having a right to their tissue. She said, well, actually, there's a surgery going on today, and we can't get it. And he asked what hospital. So he called over his surgeon general. He said, do you know anybody at X? And before I knew it, Obama was on the phone to the CEO of the hospital. 5 minutes later, I got a phone call from the general counsel of my university saying, what the happened? We just got a call from the hospital saying, oh, of course you can have it if the patient wants it. So 4 weeks ago, there was a surgery somewhere down south where, again, we consented the patient, the surgeon agreed. And what we do is this screen. So what we do is when we do this screen, anyone who works with us, any surgeon and oncologist, I officially make them a collaborator so I can share the results with them as a research result. And I agree, so I share it with all of us. I agreed with the surgeon. I would share it as a research result. Everyone was on board. 9 p.m. the night before the surgery, the family calls me up in tears saying, the surgeon just came in. Apparently the head of the whole cancer center came in and said, no, you can't give this tissue away. You want to freeze it away in case somebody in the future needs it as a control liver sample. And so the family was in tears. So I wrote to someone I know who happens to work in the White House on the cancer moonshot. I said, listen, I know you've got a lot on your plate. You're juggling a lot of balls. Here's one more. Why don't you consider something where patients can control to a certain extent where all their extra waste tissue goes, right? This is going to be an 18-centimeter tumor. They don't need the full 18 centimeters for what they're doing. 8 o'clock the next morning, the family called me up saying, what happened? The surgeon just came in and said that we can send the sample to you. What happened is the White House had called up the hospital. Unfortunately, what happened was the head of the cancer said, okay, we've got to send them a sample. They sent me a sample that was 3 millimeters by 3 millimeters by 3 millimeters from a tumor that was 18 centimeters across. He just felt he was obliged to send it because the White House said send it. I think one of the big barriers is to break down this barrier. I think that, yes, a hospital needs to keep anything they need to keep for the patient care. But when you have a tumor that big, most of the stuff gets thrown out. So my daughter's tumor was 15 centimeters. So she's the one who did the original cloning. It was at Sloan Kettering. I still have not been able to get her tumor from there. So that's one of the big barriers. Second barrier, getting medical records. So we were involved in setting up the 21st Century Cures Act, which now says that any patient has the right to get their medical records and they can't charge a lot for it. But so one of the patients just requested records that just arrived here last week, in this case from MD Anderson, and he sent me a bill for $6,000 for the records. They're bankrupting us. My wife and I have been paying for this out of our pocket. They should follow the law, which says you should make these medical records available. I mean, so those are some of the barriers that we've been dealing with.
And that's fascinating. And I mean, calling Obama may not be everyone's possibilities, but it's fascinating to hear about that. All right, Pamela.
This is Ken Dixon. I just want to interject that I very much want to reach out to Sandy after this conversation, because this is what I've been working on for several years now with our company, Specicare. So this sample acquisition is something that in the future I'd like to talk to this group about. But in the meantime, I'm going to follow up with Dr. Simon, and I just wanted to interject that.
Sandy, given those two very powerful anecdotes, have you thought of writing sort of an op-ed somewhere in some prominent place, just summarizing those anecdotes are very powerful in swaying public opinion, and I think those are very moving anecdotes, everything you just said.
So
specifically about difficulty in getting info and tumors.
So I've been trying to get the powers that be involved, because I'm not sure how much the public can sway it as much. So for example, I don't know if you're familiar with the Cures Act.
Yes, a little bit. I mean,
yeah. So we were very, very actively involved in pushing it. And in fact, what happened was so according to the Cures Act, they have to make patient medical records available to the patients. And it finally went into effect, I think, last October, November. It took like 6 years. And so we actually were so active about it when the bill was actually signed. Both Obama and Biden had invited my daughter up to actually be there on the signing of the bill. I think Biden realizes the problem. Obama realizes the problem. A few top people in Congress do. I think we need to press them to really push it through and make sure it gets enforced, and start doing the same thing in terms of patient tissue. Some kind of patient bill of rights.
I think you underestimate how public opinion can get behind these things. Like you get public opinion, social media behind you, that can create its own wave, its own pressure. I think there's numerous examples of that having happened over time. Oh, I'll shut up because I see Pam has a question.
So it's some more comments and discussion. So I first encountered this inability to get actually I could pay like $3,000 to $5,000 to get patient samples from the Northwest Biotrust, who decided that every pathology specimen at University of Washington was owned by them, and I would have to buy them back. And it was horrific. So the only way I got around that is when we had a CLIA approved test. And then what we said was that, and that we were going to return the results to the physicians, which I know you're already doing, but I was going to return clinical grade information back to the physician. And then the pathology department gave us the specimens straight. We could go to the OR, get the specimens. I just wanted to mention that because that was one thing that I encountered in Seattle. And secondly, where I am now, I'm actually Specicare is helping us get our specimens for the functional testing because I'm unable to get into the system either, but we are able to get them out through them. So I just want to mention that. Last thing I want to mention is I'm on the American Society of Hematology, and I wonder if ASCO has a similar committee on the precision medicine subcommittee. And we're like creating position statements regarding molecular testing, regarding like genomic testing. But I'm just wondering if this should be, I think that if ASCO would make a position, would create a guideline, a position statement saying that unlike the previous statements where they said everything was, I don't know, the previous statements have not been that supportive, but I'm wondering, Tony, if we could somehow work with ASCO or ASH. I can bring this to this precision medicine subcommittee at ASH, but we deal with only blood cancers. I think ASCO would be broader, where this is a huge problem because we're trying to deal with all of the issues that are preventing precision medicine advances in the clinic. And so, I mean, this is outrageous, and this cannot. I mean, I'm hearing your stories. I mean, as bad as my stories are, yours are worse, and I just can't believe how awful it has been for you.
So those are great ideas, Pam. Please continue to work through ASH. Let me know if I can help. I will say I've been working through ASCO, and at the end of this month, I'm going to be presenting to the ASCO Research Committee on exactly these issues. If you want to send me an email with some like specific points you'd like to make sure I hit, I'd be very glad to incorporate that. But I agree, I do want to get ASCO involved and work our way up through those, I think, accurate but outdated assessments of 15, 20 years ago of the inferior utility of functional assays. The technology it's a different world now, and it needs to be re-examined.
Your point about the CLIA test is a very good one. So we actually spent a certain amount of time looking into the CLIA test. So it turns out I have to put in a lot of money in terms of hiring actually 2 different kinds of CLIA lab managers because of the way the test was. And it would also be very hard to guarantee getting all samples here. So I actually fly people right to where the surgery is. I've had people fly to Ecuador, to Europe, and then bring them right back into the lab. I've realized recently that we can potentially lower the costs the following way. We've gotten to the point where, at least on this one cancer, the samples are so similar in their response that what I could do now is a very simple ELISA test for the levels of different proteins. And based on those proteins, I can predict the patient's drug response. So maybe rather than flying all the samples in, if I just get a small, when they're still healthy and responsive, from a very, very small sample, I can just do a quick test on the protein levels to say, here's the drug combination that works for this patient. But the thing is, you need to do all these initial screens to learn that work. So I think for the long term, the CLIA, is really the way to go. I'm just trying to find the most affordable way to do it because I just don't have deep pockets.
But you are returning results to the clinic, right, Sandy? You are returning results to the physicians at this time?
I'm returning it to them as a research result. And that's why in our paper, I have all these different oncologists and surgeons. I actually, in most cases, also copy the patient on it as well, explaining that this is a research result and explaining, what it is that we have. So I end up taking most of the patients in as collaborators as well. And so
do the physicians end up following your recommendation, or?
So actually, we've been meeting with the Children's Oncology Group, and they're getting ready to start a clinical trial. They've already filed with their,
fantastic.
So they're getting ready now to go to CTEP. The cancer therapy can be based on that combination. And And they've asked me to go through, for example, our early treatment was 2 weeks on, 1 week off. They asked us to do 1 week on, 2 weeks off, vary the dosage, and so the clinical trial is based on I didn't go through all the variations they asked for, but we went through and did that based on their request. And Tony, in terms of what you're saying about a sort of perspective on, I recently was invited to write a personal perspective on this whole thing, which I put into the chat if people are interested. So this was the it's the May Nature Reviews Cancer. So if people are interested. There were a lot of sort of interesting sidebars along the way on patient advocacy, on misogyny in science, on a lot of other issues that we've encountered.
Thank you for sharing that. We can probably share it with our mailing list. And so we are at 9:32, a couple of minutes past the time. I do want to note Patrick's comment on the chat that maybe we could have a working group on sample and record acquisition within the society. That could be something of interest. And we can discuss this probably next time. And thank you, Sandy, so much for sharing beautiful data and, of course, your daughter's story. And thanks, everyone, for listening. And we're going to meet again in a month from now.
Yeah, great. Thank you very much, Sandy. Thanks, Alice. Goodbye, everyone. See you next month.